Hello, everyone, and welcome to Gene Editing, Challenges and Future of CRISPR in Clinical Development, presented by Medpace. I'm Rebecca Williamson, the publisher of the Life Sciences Group, and I'll be moderating this webinar. Our speakers today are Dr. Blythe Thompson, Senior Medical Director of Hematology and Oncology at Medpace, and Dr. Trevor Walker, Associate Director of Regulatory Affairs at Medpace. You can read their full bios on the left side of your window. We've got just a few technical notes before we begin. If you'd like to download the slide deck, please click the Files button at the lower left corner of your screen. The webinar is being recorded and will be available on demand within 24 hours after the event. And we're going to be following the presentations with a Q&A session, so please submit your questions during or after the presentations, clicking that Q&A button in the lower left corner of your screen. All right, I think we're ready to begin. Blythe, please go ahead.
Good morning. Thank you very much. Trevor and I would like to thank the audience for your attendance today. Over the next hour, we will review the basics of gene editing, some recent clinical experience with the technology, followed by a discussion of the regulatory and ethical implications of human genetic engineering. The quest for editing the human genome to treat diseases has been an ongoing objective of human medicine and has evolved from a method to generate a knock-in or knockout in animals to today's precision genetic surgery for human disease. Genetic engineering is broadly defined as the process of making targeted modifications to the genome, its contexts, or its outputs. The ability to form a precise DNA break, followed by editing or correction, has been done through a variety of techniques, such as meganucleases, oligonucleotides, peptide nucleic acids, and most recently, zinc finger nucleases, transcriptor-activated effector nucleases, or TALEN, and the most recent addition, clustered regular interspaced short palindromic repeats, or CRISPR. All of these technologies have created an increasing level of interest and scrutiny. I'll focus today on three of these gene editing technologies that have most clinical data to date, specifically zinc finger nucleases, TALEN, and CRISPR, and obviously their clinical application. In brief, zinc finger nucleases are a class of engineered DNA binding proteins. They enable targeted double-stranded breaks in the DNA at user-selected locations. Each nuclease is comprised of a DNA binding domain and a DNA cleaving domain. When the DNA binding and cleaving domains are fused together, a highly specific set of genomic scissors has been created. They can then recognize independently 3 to 4 DNA bases, and when linked, can target specific sequences to bind, cleave, and either through non-homologous end joining or homologous recombination, edit the target gene. Human trials have been completed, and here is an example published in the New England Journal of Medicine of work done at the University of Pennsylvania. There was a clinical observation of a less aggressive course in select HIV patients who were heterozygous for the CCR5 delta 32 gene, and also an improved viral load in a patient who had HIV who received an allogeneic transplant from a donor who was homozygous for the delta gene. The question was then raised, if you could infuse modified CD4 T cells that had undergone gene editing, could you render that patient CCR5 delta 32 deficient? In this paper, they published their experience in a phase 1 trial with 12 patients involving CCR5 modified CD4 T cells and used zinc finger nucleases. After the infusion, the investigators observed an increase in the number of modified CD4 T cells, and 1 of 4 evaluable patients had actually undetectable HIV. The trial was felt to be safe with only 1 infusion toxicity-related SAE. Other trials are certainly underway at a variety of centers with this technology, and there are some concerns regarding off-target cleavage. The second gene editing technology that has been used in clinical trials is TALEN. In brief, TALEN are engineered restriction enzymes. Again, they cut specific sequences of DNA. The basic construct consists of a transcriptor activator effector that is bound to practically any desired DNA sequence. So when combined with a nuclease, a resulting DNA cut can occur. Clinical applications of TALEN have been published, and here is an example in Duchenne's muscular dystrophy. In 2015, Lee and colleagues published on the stem cell correction of the Duchenne's muscular dystrophy gene. The gene is 79 exons with deletions, duplications, or loss that leads to a lack of a functional dystrophin protein and disease. This large size renders traditional adenovirus-based gene therapy quite difficult. Lee et al. Used this TALEN to correct in stem cells via an exon knock-in and demonstrated proof of principle. It has been reported to have off-target mutagenesis. However, in this report, it was minimal. Of note also in this report, they actually used TALEN and CRISPR and found that they were rather equivalent. The next technology is clustered regularly interspaced short palindromic repeats, or CRISPR. I'm going to spend a little extra time because this is the newer technology describing the basics of it in bacteria and then the most recent advance published in 2012. It's been known by bacteriologists for many years and was essentially been described as an immune system used by bacteria to fend off viral infection by a bacteriophage. It was noted that there are essentially short segments of palindromic DNA, as demonstrated in the diagram in the bottom. These are interspersed with spacer DNA that are different. The spacer DNA is unique and was found to be essentially identical to viral or bacteriophage DNA. In addition to the DNA sequence, off to the left you can see the Cas protein or CRISPR-associated genes that encode for proteins that are typically helicases, unwinding the DNA, or nucleases to cut. As CRISPR is newer, we'll take a moment to describe how it works in a very average E. coli. Here's a very, very simple drawing of the basics of CRISPR and bacteria. You can see a happy little E. coli in the bacteria cell wall, the evil bacteriophage attacking at the top. As it's infecting, the bacteria is infected by the phage, the virus injects the viral DNA into the bacteria. The bacteria has seen this viral DNA before, as it's red in the image here. The Cas protein is transcribed along with the transcription CRISPR RNA, which fits into the Cas protein complex, and using the helicases and nucleases, depending on the system, of the Cas protein, breaks apart the viral DNA, and the bacteria survive. What's unique is what happens if a new bacteria infects the cell, bacteriophage infects the cell. The CRISPR system would generate a new class 1 Cas protein to essentially break apart the viral DNA. But the novel occurrence is that new spacer DNA would be then incorporated into the bacterial genome. When infected by the bacteriophage in the future, the bacteria would have essentially an adaptive immunity to this virus, using the CRISPR-Cas system to degrade the DNA. The system has been known for a long time as an immune system of bacteria, and the innovation occurred when several investigators evaluated this elegant system in strep pyogenes. In 2012, using the strep pyogenes, Dudnik, Charpentier, and colleagues described a modified mechanism to use the CRISPR system within strep pyogenes Cas proteins, specifically Cas9. The paper was published in Science in 2012 and was a revolutionary change in DNA editing, as it truly provides an elegant system that can use to precisely edit DNA. On the left here is a native type 2 system in which Cas9 is a nuclease. The Cas9 proteins constitute a family of enzymes, but they require two partners. A base paired structure formed between the activating tracer RNA, which is at the bottom with the loop, and the targeting CRISPR RNA up there in the Cas9 system in yellow. The cleavage occurs at locations determined by both base pairing complementarity between the CRISPR RNA and the target protospacer DNA in a short motif called a PAM, which is juxtaposed to the complementary region in the target DNA. We have to remember that the Cas9 system requires both CRISPR and tracer RNA. The advanced investigators then created a chimera, a combination of the entire system that effectively combines the tracer RNA and the CRISPR RNA into one chimera, essentially a guide RNA, with this linker hairpin loop. This single transcript effectively fuses the 3' end of a CRISPR RNA to the 5' end of a tracer RNA, thereby mimicking that dual RNA system that was required. And as we recall, both are required for the Cas9 to cleave. The chimera is inserted into the target cell. Cas9 will cut the DNA at the exact sequence. The cell will then direct endogenous repair mechanisms, and you could use this system essentially to remove or insert targeted DNA. What's striking about this approach is the elegant simplicity and the subsequent explosion of research that has followed. With these observations, a flurry of CRISPR activity has commenced. Dominguez et al. In Nature Reviews has provided an elegant description of these mechanisms to either interfere with transcription of a DNA, where one could see this would be example in P53, co-activation or interference in a more complex system, and finally just transcription activation. And in addition to these direct DNA applications, scientists are also investigating targeting the epigenome using a CRISPR system. And then most recently, the Berkeley group 2 days ago has published upon a RNA-based class 6 system. So certainly, we can and contemplate the myriad of applications that gene editing could be considered. In a prior slide, I discussed the zinc finger approaches to HIV. Kang and colleagues have also reported in April of this year using Cas9 to insert HIV resistance using that CCR5 into human embryos. Obviously, a natural application of gene editing would be mutation-driven cancers. And then, as we have described previously regarding Duchenne's muscular dystrophy, removal or addition of a sequence of DNA could be certainly applied to genetic diseases. Finally, the field of organ transplantation could be revolutionized in theory with possible application of this CRISPR-Cas9, even to the point that has been theorized of using mismatched or even non-human donors, which would obviously revolutionize both organ availability and human transplantation. Clinical reports are beginning to be published, and on last look this morning, there are over 4,000 publications just if you search in PubMed with just CRISPR. In 2015, Lang and colleagues reported on human zygotes where the human beta globin gene was cleaved with this system. They did demonstrate low efficiency and off-target cleavage, but have provided proof of principle. Other human trials are forthcoming, with several companies and academic centers developing CRISPR-based systems for human diseases. And their short list is listed on the slide with Editas, CRISPR, Caribou, Intellia, and multiple academic centers. In a quick look of clinicaltrials.gov, there are 4 clinical trials in human cancer that are not yet recruiting but are on their way, all in China, that as of September 2016, and actually this morning, we're not recruiting, but we're targeting a variety of human malignancies, all using CRISPR technology. And here in the United States, the University of Pennsylvania has received RAC approval for the first U.S. trial using CRISPR-Cas9 technology. They're targeting a combination of malignant mutations, PD-1, NY-ESO, and LOG1, but in a human cancer. Gene editing technologies has limitless applications, from drug development, disease models, ecological viral control, biofuels, and most recently, reported in Sweden, even modifications of human crops. In this case, a CRISPR-modified cabbage has been produced and eaten, and most recently, there's a CRISPR-modified cucumber. Advances in CRISPR technology are continuing, and as I mentioned just 2 days ago, we've now moved on to an RNA type 6 CRISPR system at the University of California in Berkeley. As a medical director at a clinical research organization, it's astounding the speed at which CRISPR-Cas9 has gone from bench to bedside and the global reach of this technology. We have to wonder if the speed of clinical trials and regulatory review will keep pace, as we remember this was just started in 2012. To discuss the regulatory and ethical path forward, I'm going to pass the controls to my colleague, Trevor Walker, and a reminder at the end we will have time for questions.
Thank you, Blythe. Okay, so as Blythe has mentioned, I'm going to discuss some of the regulatory and ethical implications of the CRISPR technology and how it relates to clinical development of new therapies. As Blythe has outlined, there's a myriad of uses that the CRISPR technology can be used for, and in particular for the development of innovative therapies that do have the potential to provide a lifelong cure following a single treatment of one of these therapies. And that's hugely attractive for patients, especially where they're suffering possibly a progressive degenerative disease. The option of taking a single treatment versus a lifetime of ongoing drug treatment is certainly highly attractive, and the public are keenly watching developments in this area. CRISPR technology has certainly made gene editing much more accessible. One of the attractions is it's technically more straightforward than the other types of gene editing technologies. The precision of the editing has certainly improved, and there's been a reduction in off-target effects, and that's certainly a great advantage. We're becoming very well aware that the potential for clinical development is going to be vast as this technology opens up the whole of the genome to find potential targets. And the CRISPR technology is going to allow this search for suitable targets to progress much faster than ever before. For successful clinical development, what there will need to be in place are robust regulatory and ethical frameworks to ensure that product development meets the required quality, safety, and efficacy standards that the regulators need. The challenges for the developers include navigating the current regulatory pathways in order to be able to progress their products to reach the patients and meet the unmet clinical needs that are there. These will also be time sensitive to bring these effective therapies, especially the ones that display early potential, to the market and to the patients as early as possible. The regulators themselves are already aware that early access to medicines is important in the development of new therapies, and especially for those innovative technologies. So it's essential that a safe and rapid regulatory pathway can bring these CRISPR-based technologies to the clinic. Key to this is going to be the early engagement of developers with the regulators, and achieving an open and constructive dialogue at a really early stage and right through the development pathway is going to be critical. So the use of CRISPR for gene editing in human cells will be primarily used for the development of somatic cell therapies and could also be used for the development of germline therapies. And in this slide, I've got a few side-by-side comparisons for these two types of therapy. So somatic cell therapy involves changing the DNA in specific somatic cells of an individual. And that treatment to modify these cells will result in changes in the genetic makeup, and that could be reflected at the protein level. Ultimately, when the patient dies, these modified cells will also die, and treatment of somatic cells won't result in the passing on of any of these genetic modifications to future generations. It's anticipated that somatic cell therapy is where the main focus will be for clinical development, and this is what we're seeing with these initial clinical studies that are already being proposed and starting to be initiated. Somatic cell therapy produced using CRISPR technology is going to raise the same ethical and regulatory concerns as have already been raised with current gene therapy procedures. So the existing regulations and guidelines that provide the requirements for development of safe and efficacious products, as well as considerations to provide positive risk benefits, will govern these applications. We don't anticipate that there should be any significant ethical concerns or issues for somatic cell therapy. And as you can see already, basic research is advancing and therapies that are going to move to the clinic are rapidly being developed. In terms of germline therapies, this is gene editing where germ cells or zygotes are edited. These types of therapies raise more challenging ethical and regulatory considerations, and this has certainly sparked significant debate and discussion globally. Gene editing of the germline would produce changes in the DNA that can then be inherited by subsequent generations. It's feasible that intentional germline editing for serious genetic diseases could be carried out at an early embryonic stage using the CRISPR-Cas system, and would then result in correction of mutations in all the cells of the person that developed from those initial zygote, or that initial zygote, and that would also include gene modification of their germline cells. So what it would mean is that a specific gene-edited modification could then be passed on to subsequent generations, and that could potentially remove a serious genetic disease from a whole family. So you can see that there's a very positive effect that could occur for germline editing. However, germline editing does raise a number of significant ethical issues. Gene editing of the germline would result in, as we've said, introduction of modified genes into the human gene pool, and it could be difficult to remove these if it was subsequently found that there were additional harmful effects. There is the risk of inaccurate editing. It may be difficult to predict harmful effects as a result of gene editing ahead of time. There could be implications for the individual and also for future generations who carry that mutation through their germline. And in terms of impacts on altering human evolution, these are also serious considerations. What's also feasible in terms of germline editing is the use of this technology for non-therapeutic use and allowing modifications of the genome which would lead to enhancement of traits or abilities within individuals. And this also has significant implications for society as a whole. At the moment, there's inconsistent and variable regulation on germline therapies across the countries, and these range from legal prohibition in some countries, it's restrictive in other countries, and in other countries the legislation is very ambiguous. Currently, there is basic research of germline editing allowed in early stage embryos, and that's certainly been seen recently in the UK. This type of research is legislated, and it's legislated such that it specifically states that the basic research can be carried out, but it cannot be used as the basis for a therapy. So there are already some legislative restrictions in place. As a result of these ethical concerns, and also a number of technical challenges, it's expected that the development of therapies involving germline editing are likely to be restricted. Just a few more points on the concerns around non-therapeutic use or enhancement. There was a recent meeting, an international summit on human gene editing in December 2015 to discuss the development of CRISPR technology and its ability to make gene editing more accessible, and also to discuss a number of the concerns that the potential use of this technology could have. The outcome of this meeting in December was a statement that was released, and some of the key points that were addressed were that basic research, including preclinical research, would be appropriate to continue, but there would be a requirement for appropriate rules and oversight to be in place. It was also noted that clinical use of somatic cell therapies would certainly be acceptable. However, concerns around germline editing were raised once again. It was accepted in principle that there may be cases where, for severe inherited diseases, there may be a case for germline editing, but that the risks that this therapy could be used for enhancement of human capabilities provided such a great ethical concern that at the moment it would be recommended that a moratorium was put in place. There was certainly a recommendation that an ongoing forum to try and establish acceptable uses for germline editing was set up, and also they should try and look at harmonization of regulations, and that should be done at a global level. A subsequent meeting in Paris in April this year had a specific focus on a number of aspects of human germline editing, looking at the consequences, regulation, potential applications, and again, concerns over the capability for this technology to be used to enhance specific traits in humans were raised. In parallel, there's also a multidisciplinary committee has been assembled, and they're going to perform an independent year-long in-depth study to look at the science and the policy of gene editing, and that will provide a very vital report on the future of any germline editing developments. So a number of countries, including the US, have requested a moratorium on using gene editing to make heritable changes to the genome, and also to explicitly stop the potential for non-therapeutic use. So this is going to remain a key issue that's going to be debated for the coming few years as the clinical development of somatic cell therapies continues. In terms of existing regulation for germline editing, certainly the European CT directive does not allow germline editing, and also the NIH Recombinant DNA Advisory Committee will not review proposals on germline editing. The concern over enhancements has been raised certainly at all of these meetings, and there are a number of conventions, these are international conventions already in place, that will restrict any practices that would allow non-therapeutic use of gene editing that would lead to enhancements and could potentially be considered as eugenic practices. So already there are some international regulations in place, but it's felt that these may need to be revised and added to. So now to focus on somatic cell therapies, and as we mentioned, these are the most likely to be where we see the fastest developments and will result in the biggest impact in treating clinical conditions. These therapies are going to use either an in vivo strategy to target cells and tissues within the body, and will rely on efficient and safe delivery of the CRISPR-Cas components to the target cells or tissues. The other alternative is using ex vivo strategies, where the cells are removed from the patient, engineered, and then administered as a therapeutic agent. The advantage, of course, of ex vivo editing is that it would then allow the cells to be selected and analyzed post-engineering, and it would mean that the cells that were then used for therapy would be the correctly edited cells and would potentially provide a relatively pure population. The current regulations in place for cell and gene therapies are going to be used to regulate the development of CRISPR-based therapies, but the regulators will need to ensure that they stay aware of the rapid advances in this technology. It's anticipated that the regulations and guidelines will need to undergo regular review to ensure that they're keeping up to speed with the developing technology advances and be able to provide appropriate guidance as this technology moves forward. And most importantly, the regulatory pathway from development to the market will need to be flexible while it still ensures efficacy and safety, and also be able to allow timely access to these novel and innovative therapies. So as with all therapies, demonstration of safety and efficacy is one of the main focuses for the regulators. As we mentioned, the current gene therapy guidelines will be applicable for these therapies, and they already provide information on expectations and requirements. One of the main concerns with gene editing, and particularly for CRISPR, is the potential for off-target effects, where cleavage or editing of unintended targets could result in unwanted activation, inactivation, or disruption of genes with subsequent serious downstream impacts. The potential for off-target effects and genotoxicity have been addressed by developments and improvements in the CRISPR system. So refinements to the system have certainly improved specificity, and there's been a reduced occurrence of off-target effects. And this area we think will remain as a specific area for further development as these therapies move through the clinical development pathway. Regarding detection of off-target effects, there are now methods available to allow genome-wide analysis, and these methods can detect off-target cleavage with sufficient precision, resolution, and sensitivity that they should be able to provide evidence to objectively determine whether any off-target editing has occurred. So this ability to assess off-target effects across the whole genome will help to demonstrate specificity and also, importantly, safety. Another key safety aspect is the ability to show that there's going to be no detectable modification to the germline following treatment. And that's obviously not intentional germline editing, that would be unintentional germline editing, but it's important to show that that's not going to be a risk factor. So that addresses some of the aspects around safety. In terms of efficacy, CRISPR-based therapies, they have the potential, as we said, that a single treatment could provide a lifelong cure, and that's depending on the treatment strategy. So in particular, for in vivo somatic cell therapies, where complete knockout of genes in a cell population is required to produce the required clinical effect, there may be the requirement for multiple treatments. And with that, there comes some concerns around treatment-associated immune responses, and that may in part be due to immune responses raised against the vectors, such as adeno-associated virus. The other aspect is regulation of the editing system itself. So the ability to have a controllable switch to either activate or deactivate the CRISPR components is an important development. And already we've seen these types of developments whereby UV light or drug-induced switches has already been developed, and this is going to be important in the regulation of the CRISPR system. It's going to be very important for the regulators, and again will provide additional safety and efficacy confirmations. Another aspect to consider is around the non-clinical testing in animal models. This is always a key area of focus in all cell therapy and gene therapy products, and particularly for the development of CRISPR therapies. There'll be a real focus to provide sufficient safety and efficacy data prior to any first dosing in humans, just to confirm that there are no untoward risks that could be associated. Selecting the appropriate disease model always represents a challenge. That disease model has to closely represent the clinical condition that's going to be tested. One of the other considerations, and this is for the wider drug discovery spectrum, is that where it's been difficult in the past to develop a specific knockout animal or a cell line with specific genes that have been knocked out, the potential of CRISPR-Cas technology is that it can be used to develop these model systems because of its specificity. then it can be used to create models with the correct mutations. And this, we believe, is going to have big applications for that much wider spectrum of drug discovery. So there's huge potential here, not only in development of CRISPR-based technologies itself, but also in support of other areas of drug development. Now, just a few points on quality, manufacturing, and administration. So due to the nature of these types of CRISPR gene editing therapies, the overall manufacturing process is likely to be time-sensitive, labor-intensive, expensive, and technically demanding. And those requirements will need to be met in order to achieve the development of a robust process that meets the quality and consistency that will be required from the regulators. Depending on the product, release testing may need to rely on rapid testing for attributes such as sterility, mycoplasma, and endotoxin, and particularly where there's a time-sensitive aspect to release of the product. Some of these challenges are already relevant for other types of cell and gene therapy, and there are appropriate strategies and assays that will be applicable. Another aspect that needs to be considered is where there is a short shelf life, and this could be in the matter of hours rather than days, weeks, or months. So there can be additional challenges associated with this, including geographical location, storage and in-use stability data, transportation logistics also may need to be factored in, and where the administration site is located is going to be key. If a product's been developed and can be cryopreserved, then it does give the potential that a global supply could be provided from a single specialized manufacturing site. Otherwise, developers are going to have to look to setting up regional manufacturing options if they want to be able to target a global market with their product. What we also expect to see is that there's going to be an increasing requirement for patients to attend specialized centers for treatment, especially where CRISPR-based therapies are based on autologous cells. And in these situations, there would certainly be advantages for manufacturing to be based at the sites of administration, or at least close to these treatment centers to minimize any storage or shipping logistics. And the other point to raise is we're already aware of stem cell therapy tourists. And I think it's likely that with public awareness around the potential of the CRISPR technology and the resulting therapies that are going to be developed, there's going to be a significant increase in patients who will be prepared to travel globally, not only to participate in clinical trials, but also once these products are approved, they'll be prepared to travel to receive treatment. Here I wanted to raise a few clinical trial considerations. In the US, Blythe has already mentioned the Recombinant DNA Advisory Committee. The role of the RAC has been streamlined in 2016 following some changes to the process, and the hope is that fewer gene therapy studies will go through public review. But as we've seen from the very first CRISPR study that's been approved by the RAC, because this is a new technology, there's likely to be a requirement that subsequent studies involving CRISPR-based technologies will need to go through public meetings. The current process for RAC public review is that the protocol will be reviewed by an IRB or an IBC, and they will request a RAC public review if it meets certain criteria. The impact of this is that it will lengthen the timelines to initiate clinical studies. But what it does do is, in these early days, it ensures an appropriate level of oversight and review of this technology. In Europe, gene therapy studies do have additional time for review. That's already built into the European Clinical Trials Directive, and it may also involve additional review experts where required for specific innovative technologies. The other thing to consider is where there's the use of modified viral vectors. These are considered as genetically modified organisms in Europe, and where these are used to transfer the CRISPR-Cas components to the target cells. These will require additional authorizations and approvals from European countries. One of the challenges in Europe is that currently there's a lack of process consistency, requirements and timelines to obtain these GMO authorizations. And many stakeholders have already raised issues with this divergence across Europe and are requesting that there's resolution and some consistency brought across the requirements. What we expect is that as the regulators gain experience with the CRISPR-based technologies and how they're going to meet the requirements for quality, safety and efficacy, they'll gain a certain level of comfort with the technology. It may be that there's going to be an increased focus on review by ethics committees and IRBs when clinical trials with CRISPR-based technology therapies are submitted for review and approval, and potentially there could be, for some time to come, additional questions raised, and this will impact the startup process for clinical trials. Looking at the regulatory pathways to get products to market, certainly these need to be timely and flexible, and faster access to developing therapies would certainly be an advantage. Within Europe, Japan and the US, there are established pathways that can expedite regulatory approval and placing of products on the market. These pathways are designed to ensure that safety and efficacy are still demonstrated, and this can be in the form of additional safety studies that are required to be completed within specified timeframes as part of a conditional approval. Within Europe, there are the adaptive pathways that's based on the pre-existing conditional approval system. More recently, the PRIME initiative for priority medicines is available, and accelerated assessment is in place within Europe. Japan in 2015 made revisions to the PMD Act, and they have in place an expedited approval system for regenerative medicine products. And they have the ability to provide a conditional approval for a product, and within 5 years the developer needs to complete the additional studies that would be required for full marketing. And then within the US, there are the accelerated pathways. These include fast track, breakthrough therapy, accelerated approval, and priority review. And these all focus on moving forward products that can meet a high unmet need in patients. Moving on now to the cost and reimbursement challenges. As seen with a number of the cell and gene therapies that have already been approved in Europe, CRISPR-based therapies are likely to have a high cost per treatment. And coupled with the fact that some of these therapies will be targeting rare diseases that will only have a limited number of patients, this is going to be a challenging combination from a financial perspective. Certainly some advanced therapies that have already been approved in Europe have found it difficult to make these therapies profitable or even to actually meet the manufacturing costs, and at least 2 of these therapies have been suspended. One thing to consider is that a single one-off high-cost treatment may be more cost-effective than a lifetime of existing drug treatments, but there may be a reluctance to pay these high costs until long-term safety and efficacy are demonstrated for these therapies. Developers and HTAs are going to have to resolve these issues to ensure that these therapies do in fact reach the patients with the diseases where there's a high unmet need, and that the full potential of CRISPR is realized. What's interesting to note is that in terms of new approaches to pricing, Strimvelis, which is a GSK product that was recently approved in Europe, they have a pay-for-performance agreement in Italy. And perhaps for some conditions and some therapies that are developed, this might be a potential solution. So As we've outlined in this presentation, the expectations of CRISPR-based therapies is very high, and it's anticipated that this is going to be the focus of development for a number of companies in the coming years, and certainly there's a vast increase in the use of CRISPR technologies, certainly at the basic scientific research level as well. It's expected that the predominant focus of clinical development is going to be around rare genetic diseases, mutation-driven malignancies, and cardiovascular indications. And really, the potential for this technology is vast. As we've mentioned before, really now the entire genome has become full of drug targets, and the applications really have the potential to be limitless. As we've stressed, a flexible approach to regulatory pathways is essential, and developers, ethicists, and regulators need to be discussing and collaborating at a very early stage. And certainly regulators continue to confirm that they're more than happy for developers to reach out to them as early as possible and have discussions throughout the development pathway. And that way, it's going to ensure that these therapies actually make it to the market and prove their efficacy and safety. And really, ultimately, the long-term efficacy and safety is going to demonstrate the value of these technologies and also their potential to treat complex and challenging diseases. I'd just like to wrap up with this extract from a recent review on CRISPR gene editing. And this highlights that the development of gene editing therapies holds great promise, but it's also going to rely on careful development and appropriate regulatory control for these therapies to realize their full potential. Thank you for your attention today, and I would like to hand back to the moderator.
Okay, great. So now it's time to move on to Q&A. I want to remind everyone there's still time to submit your questions using the Q&A button. Again, it's in the lower left corner of your screen. So we'll try to get to as many questions as possible. Bly and Trevor, do you want to go ahead and take away the Q&A?
Sure. Thank you very much. And I would like to thank the audience for their attention, and we've received lots of questions. Briefly, I just wanted to mention that both Trevor and I will be, if you want to talk to us in person, because I don't know if we'll get through all these questions, we'll be at the Alliance for Regenerative Medicine Cell and Gene Therapy meeting next week at La Jolla, California, and happy to talk in person, but because there's a lot of questions. So I'll go through these and pass it back and forth between Trevor and myself. There's been a lot of questions asked about regarding immune mechanisms and immune reactions to CRISPR-based therapy, and I think it's a great question, and certainly when adenovirus might be used as a delivery vehicle. I think right now we are very much in the nascent stage of this clinical development, and absolutely in clinical trials as an oncologist, we are highly recommending that it's done very safely in phase 1 trials and absolutely monitoring for immune mechanism, especially wherever it's being delivered. And I think a lot of our guidance could come from our gene therapy colleagues where they have seen, where they've been monitoring and using adenovirus-based technologies. So a lot of our knowledge will come from these gene therapy-based drug delivery systems. But absolutely, I think it's all too new for us to actually know what will be the immune mechanism, what will be the immune reactions if you'd inject it directly into a lesion and directly into a particular body part. I think it may all be a little bit too new to tell. Trevor, do you have any other, obviously you had mentioned during your presentation about monitoring for immune toxicities. Any comments or thoughts on immune-mediated toxicities?
I mean, I think you're quite correct, Blythe. I think that we can potentially take some information from what we've seen in the existing gene therapy knowledge that's out there, and especially for AV or AAV based vector systems. but it's certainly going to be interesting to see whether there's concerns over immune responses. And I guess part of it is going to remain around whether a single treatment is going to be sufficient to deliver a required amount of gene editing, or whether subsequent treatments are going to be required. And it's where subsequent treatments are going to be required that may flag up potential issues.
So another question has been asked by the audience about, and Trevor, you could perhaps theorize on this, which country do we think, the United States or in Europe or elsewhere, would be where you would consider to approach for clinical applications of somatic therapies? What territory would you advise starting? Do you have thoughts on that, Trevor?
I mean, that's a very good question, and obviously what we've seen in terms of the studies that you mentioned, Blythe. There's obviously a study from the University of Pennsylvania is in the process of being approved and initiated in the U.S., and I think that there's a good regulatory pathway in the U.S. for that. It's interesting that clinicaltrials.gov has 4 studies that are pending recruitment. Those are in China, so it's going to be interesting to see how those develop. I think within Europe there's a strong basis of regulation and guidelines for gene therapies, and as we mentioned, for somatic therapies, they're going to fall under the gene therapy regulations. So I think that certainly within Europe, these types of therapies would be well received, and I think there's an appropriate regulatory framework there to review those and approve clinical trials.
Just along that line, as we contemplate this, several of the audience members and myself wonder if we ever all could get along, and whether there ever would be a global regulatory review, and whether all of the countries are interested in it. Specifically, several audience members have questioned about China, whether they would participate in global regulatory overview. Do you think that is, Trevor, do you, in your opinion, think that that's something we could ever get to, or that would be way too far in the future?
I mean, in my opinion, I think that's quite a long way off in the future. I mean, certainly the ethical concerns around germline editing, we may reach an international consensus on that, but I think for somatic cell therapy that's developed through the CRISPR-Cas system, it would be nice that a global information or regulatory package could be used in all of the major regions, but I think that's still a long way off, and we're still going to see country-specific requirements and certainly regional-specific requirements in Europe that are going to differ. And I think that would be an ideal situation if we could move forward to a single global quality, safety, and efficacy package. But yeah, I think unfortunately that's some way off, and I think that the developers are really going to have to decide whether they want to focus on one particular region that they're going to develop their product, or if they're brave enough to go ahead with a global strategic approach, then they probably just need to accept that there's going to be variations across the regions, and they'll just need to deal with that as appropriate.
Another question has been raised by several people about off-target effects of the CRISPR-Cas9 and other systems. And certainly, I think again here, the technology, the clinical experience, is really not known. I think the large concern of off-target effects would be you will hit the wrong portion of the genome and get a mutation where you had not planned and actually develop either disease or sequelae of that off-target effect. There's also been questions raised about when selecting guide sequences, to what extent are these databases guarding against off-target gene editing? And I think, again, that's reliant upon it is exploding rapidly, and we're somewhat reliant upon regulators looking at the animal models, preclinical models, as best they can, and then obviously doing deep dives into the sequence. But that preclinical experience, as you bring it to a human, would certainly, hopefully, give you an idea what the off-target effect might be. But this is quite new. I don't think anybody can actually know, especially in somatic therapy, what the 10, 20, 30-year effects of an off-target effect might be, that may not even be visible for 10 years. And certainly, again, I go back to our gene therapy colleagues where they are monitoring for off-target and insertional mutagenesis. I think there'll be a whole new nascent field about off-target effects and monitoring long-term and longitudinally for the patients who undergo any CRISPR or gene editing technology. Trevor, do you think that, when we contemplate the regulatory approval and long-term follow-up of the patients who undergo this, do you think that taking the lead from our gene therapy colleagues would be reasonable, or should we start looking elsewhere?
No, I think that's taking the lead from the gene therapy colleagues is going to be the place to start. Certainly for gene therapy at the moment, there is a requirement for long-term follow-up, and I think that's going to be key and really will identify the potential and the efficacy for these types of therapies. So I think there's a lot of reason to gain information and knowledge from what's gone before in the gene therapy field, and to use that to our advantage in developing these therapies.
Another question has been raised about the integration of targeted patient organizations in CRISPR development. And I think I absolutely agree that in our current era where patients are truly advocates and need to be involved in, especially patients with rare diseases, I think that we need to listen to the rare disease population and work collaboratively. They will, as a clinical research organization, we absolutely reach out to our rare disease patient advocacy groups, because those are the patients who are being affected by this. Those are the patients who are on the front lines. And absolutely, we must work with patient organizations for both ethical, clinical, and long-term effect, and answer what the patients need. I think there's another question about where do we potentially see the first technology or the first CRISPR application. I think we've already seen it. So obviously, when we were considering any clinical trials, we weigh the risk benefits. Those patients who have life-threatening diseases, specifically cancer, you can see that the original trials are all very focused on oncology. I think also the patients who have rare diseases for which there are no potential therapies, those are the patients who also would be those true orphan diseases, the rare diseases that have currently no effective therapies, that have clinically significant diseases. Those are the patients who are going to the trials where we're going to see the CRISPR technology immediately applied. But obviously, and then finally, as we are reading in the popular press, in vitro fertilization, and there's been investigators in Sweden at the Karolinska Institute who's also considering doing preclinical investigations in the application of CRISPR technology in IVF to figure out why particular families cannot conceive. Trevor, do you think that that would be a sort of a reasonable supposition as to where the first wave of applications would apply?
Yeah, I mean, I think that we're going to see a huge targeting of oncology indications, and I think those are likely to be the ones that are going to have the biggest impact. And there's certainly going to be developers out there who are targeting specific rare conditions where the genetic mutation is very well identified and there's a very clear target. And CRISPR now gives us gives them the technology and the tools to target those specific mutations. So I think the short term it's going to be oncology indications, and I think following on from that we're going to see a big impact on some of these rare conditions.
The ones that have very defined mutations that are not been amenable to prior adenovirus or other therapies.
Absolutely.
Yeah,
absolutely.
An interesting question has been asked whether there's a risk of do-it-yourself gene editing due to the ubiquity of CRISPR, and how could we control that? Absolutely, these cassettes are becoming commercially available, and I think that it may be very hard to control the application of that. Trevor, do you have any comments on how we can prevent do-it-yourself gene editing?
I mean, that's a very good question, and it points to the fact that this technology has become less technically demanding. The cost has dropped. There's increased specificity, so the potential's out there. But yeah, how that's regulated and controlled is a very, very difficult question, and I really don't know how the authorities could begin to address that. That's going to be a tough challenge.
Yeah. So, Trevor, I think we're all done.
Okay. Thank you.
All right. Well, thank you. Oh, sorry, go ahead.
No, I was telling to stay.
Okay, great. Well, thanks so much. Again, that was our final question. And we had a lot of great questions and wanted to remind everyone we will get back to everyone submitted personally after the webinar. I want to thank you all for attending the Fierce Live webinar, again, submitting so many great questions. I'd really like to thank our speakers for presenting and Medpace for presenting as well for today's webinar. The webinar has been recorded. You will be able to access the recording within 24 hours on the same page that you used to register for the event. So thanks again for joining us, and we look forward to seeing you at future events.